So I'm gonna explain the tyrosine kinase signal transduction pathway. So you have two tyrosine kinase receptor proteins, and you have these signaling molecules that attach to both of the receptors, and this attachment activates them to squeeze together into one dimer. And now that it's activated, it's going to use the ATP floating around randomly in the cytoplasm. It's going to take off a phosphate, and this side will phosphorylate this side phosphorylates this side, until you have all the inner receptors full, and it's fully phosphorylated. And now a relay protein is going to come in, and it changes its shape to fit around the phosphate. So the shape changing will create a cellular response, because the conformational change activates the relay protein. And so here, this activated relay protein is going to activate one of the protein kinases. And now that you have an active protein kinase, it's going to activate another one. And now that this one's activated, it's actually going to take up a phosphate from some of the ATP that was floating around randomly in the cytoplasm, and it's going to become activated with a phosphate now. So it changed shape, now it has a phosphate. So then this active kinase is going to activate this one, and then this one, now that it's activated, it's going to take up a phosphate and have a weird shape change with a phosphate. And now the phosphorylation cascade comes to an end when this active kinase activates this protein, this different protein. So now that there's an active protein, it's going to create a cell response, and this cellular response will activate this transcription factor. So now we're in the nucleus, and now that this transcription factor is activated, it's going to sit on the DNA, and it's going to tell the DNA which genes to write an RNA strand from. So it's going to take certain genes from the DNA, make an mRNA, and then the mRNA, it's going into the cytoplasm now. And in the cytoplasm, it's going to go into a ribosome. And when it goes through the ribosome will put amino acids next to each other to make the protein. And you need 3 mRNA nucleotides to create 1 amino acid.